nanometer, 1/1000th of a meter, the scale at which nanoscience of biomimedics starts. 1 nanometer. It is the size of chemical molecules biomimedics researchers work with every day, but also of the molecules of the nutrition you've had for breakfast, like for instance glucose. 1 nanometer is also the length your fingernail grows in just 1 second. The nanometer size scale is where you will find the BioMIMedics nanoparticles, with dimensions resembling viruses. With their nanometer sizes, these nanoparticles are much smaller than the smallest living building blocks of our body, our cells. Compared with a red blood cell, which has a diameter of 8 micrometers, the BioMIMedics nanoparticles are 190 times smaller. This equals the difference in length between the famous all-time basketball player Michael Jordan and the Empire State Building. Within the BioMIMedics consortium, researchers from all academic R&B centres collaborate cross-border on the design and synthesis of these high-tech chemical nanoparticles and the analyses of their behaviour and function in living cells and preclinical models. The novel chemical models created by BioMIMedics can be orientated and assembled into unique nano-sized materials of various shapes, like little Lego blocks. These chemical Lego blocks self-assemble in water or oil-like solutions into linear nano-sized structures or sphere-like beads that are hollow or contain highly porous structures. At the CERN Institute of the University of Liège, researchers have been working on very special nanoparticles called micelles. These micelles are composed of novel chemical molecules that are based on the molecules that can be found in our body's DNA, called polyphosphoroesters. BioMIMedics has shown that these polyphosphoesters are an excellent class of new polymers that are highly biocompatible and suitable to design all types of medical applications, like for instance, drug delivery.
For the BioMIMedics project, we developed in our laboratory the synthesis of novel biomedics polyphosphate-based nanoparticles, and we investigate their potential as drug delivery system. Indeed, a nanoparticle is composed of an hydrophilic shell and an hydrophobic core. And in this hydrophobic core, it's possible to encapsulate an hydrophobic drug. By modifying the nature of the hydrophobic core, we can modulate the parameter of the micelles, such as the size, the loading capacity, and the release profile of the drug into the body. As biomedical applications are foreseen, we investigate also the toxicity of these different nanoparticles in aqueous media in our consortium.
At the lab of RWTH Aachen, DWI BioMIMedics researchers focused on new technology to create silica-based nanoparticles with high efficiency and by using eco-friendly materials and solvents. Silica particles of different shape and sizes can be created during their production.
In the framework of BioMIMedics, we have developed a new technology, a surfactant-free technology, to encapsulate different substances in silica microcapsules or nanocapsules. And we work very closely together with our partners to use these particles, for example, for diagnostics as well as for drug delivery.
Like little pieces of art, these silica particles present remarkable shapes and composition. How do these nanoparticles work in, for example, medical applications? When these nanoparticles are equipped with a landmark tag and filled with a drug, they will, upon injection into the body, only bind to specific cells inside the body to deliver the drug. For example, cancer cells within a tumour located in the liver. At the level of the tumour cells, the nanoparticles bind specifically to the diseased cells, after which they enter the cell and deliver the drug that will kill it, and thereby, eventually, the tumour. It is almost magic, nanoscience. Chemistry and biology at its very essence. Mimicking nature, delivering materials inside cells in order to change the larger picture, to keep you healthy. On the level of imaging and testing the cellular behavior of nanoparticles and preclinical efficacy, BioMIMedics has created a strong infrastructure and R&D toolbox fit for now and the future.
So our system allows to visualize pretty small particles, so-called nanoparticles within the cell. And this is important to find out how the drug delivery released by these nanoparticles actually can be visualized and traced with respect to the behavior within the cell. So our method, called image correlation spectroscopy, allows to monitor the dynamics and the fate of the nanoparticles within the cell. So we can look really at the dynamics itself without paying too much attention to the localization. Of course, localization is important. The localization with organelles such as mitochondria and lysosomes is important, but of particular interest is the dynamics of the nanoparticles and the released drug. So therefore, we are spending a lot of energy concerning data analysis, image interpretation concerning dynamics of nanoparticles within the cell.
Another fine example of BioMIMedics neuroscience is the patented linear polycation polymers created by BioMIMedics researchers at the CEIB lab at the University of Liège. These unique polymers hold strong potential to become a groundbreaking alternative in daily medicine. In current medical practice, a frequently used drug to prevent blood clotting during cardiovascular surgery is heparin. At the moment, only one antidote is available, protamine. Although effective as a heparin antidote, protamine is a product that can come with severe side effects.
So the idea was to replace this product by a synthetic antidote and a polycation ones. So the positive charge of the product, so the polycation, will be able to neutralize the negative charge carried out by the heparins. We use a polycation for a medical application and not anymore like a medical device, but a real drug. But to go further and to arrive to launch on the market this product, we need, of course, a lot of other experience. We, of course, are looking for new grant and new project to go until the end and to finally succeed to launch the product.
BioMIMedics also investigates, together with companies in the EU region, the options for nanoscience in their daily R&D and business. One example is the innovative research company CRB for cosmetics and skincare products, located at the Brightlands campus in Maastricht.
4 years ago, we decided to invest here in Maastricht to be more close at the basic research activity from the university. And after 4 years, I can tell you, we are entering in an interesting collaboration with BioMIMedics. We find beautiful new materials, and for that we can really create a breakthrough new product for the market.
Pick your application: delivery of a drug to the lungs, heart, brains, or a new formulation of a long-lasting sunscreen delivered to your skin by using nanoparticles, or developing novel antibiotics. It's all possible with smart nanoparticles. Our next goal? Bringing it to the market. A challenge that requires a strong EU-regional private-public R&D network, infrastructure and investments, for now and the future.